everyone, and welcome to today's Kneat webinar, the 3 stages of process validation. As we allow everyone to log in and get settled, I just want to go through some of the housekeeping items you can see on the screen. We definitely recommend using earphones or a headset to listen to the event, as the audio is the best sound quality that way. You are muted during the presentation, but we encourage you to ask questions throughout. There is a question and answer box on the right-hand side in that GoToWebinar control panel. Feel free to type those throughout the event, and we'll get to as many of them as possible at the end. If we do run out of time for your questions today, we will make sure to follow up in an email directly. There also will be an email sent out later this week or early next week that will include the slides and recording of today's presentation, so you'll be able to go back and reference it if you need to. If you have any other additional questions about this webinar that come up after the time, feel free to always email us at info@kneat.com. We always appreciate your patience. All right, so moving on to the next slide. As mentioned, we are looking at the 3 stages of process validation with speaker Olivia Calder, who's one of our process validation subject matter experts here at Kneat. The next slide is just our privacy statement. We always share this before jumping into the presentation. But with that, I will bring it up to Olivia to introduce herself and get started with the presentation. And we hope you enjoy it, and we'll leave some time for some questions at the end. Oh, I spoke too soon. There's one more slide. For any of those on the line that are maybe not as familiar with Kneat, we are a public company listed on the TSX. We now have, we're actually close to 300 employees now. We are used by 8 of the top 10 big pharma companies, and we have over 60 customers that are global. 40,000 users are in the Kneat SaaS platform, including 150 site global deployments at different companies. We have a certified global partner network with over 40 organizations, and we operate in Switzerland, the US, Canada, and are headquartered in Limerick, Ireland. All of our software is developed in-house, and we are ISO 9001 and 27001 certified. So just a little bit of information about Kneat before getting started. On the next slide, there is a picture of Olivia, who you can see today. She is our subject matter expert in process validation and will be driving on with the presentation today. And as mentioned, yes, you will get a copy of these slides following the presentation today. So enjoy the webinar, and over to you, Olivia.
Thanks, Madison. So hello, everyone. Thank you for joining today. My name is Olivia Calder. I am a process engineer here at Kneat in the professional services group. Prior to Kneat, I worked for a pharmaceutical contract manufacturing organization where I was a process validation engineer there on the technical transfer team. So most of my clients at my previous role were drug product related, although I did work with some drug substance clients as well. So as we go through, you might notice that a lot of the examples are based off of my drug product experience with a few drug substance things here and there, but do keep in mind that all this information does pertain to other industries as well with drug substance and med device. But with that, we'll go ahead and get started. Right, so to start us off, I did want to begin with what is process validation, just so we are all on the same page here with different definitions. So all the definitions I have up here are from the Guidance for Industry for the General Principles and Practices. I know sometimes when you see a definition, it might seem a little daunting, so I will make it into simple terms, and then as we go through, we'll go through each of the stages in more detail. So first off, for what is process validation, the big question. So in simple terms here, the goal of process validation is to prove to the FDA or the EMA, wherever you are in the world, the agency will depend on your location, that your process is under control and you are delivering consistent quality products. So that is the big goal of this, and then all of these 3 stages actually pertain to that goal. So you'll see that as we walk through. So for stage 1, this is process design. So in this stage, research and development will take place, and scale-up activities will also take place. You will basically gather those initial data points in order to start to form your process from start to finish. So you're getting to know your drug a little bit here and gaining confidence based on that data that you find to then go into stage 2. And then stage 2 is process qualification. So many of you might be familiar with this stage if you've worked with the CMO or you have experience in that industry. So this is where our PPQ batches will occur, which is short for process performance qualification. So you're taking all that data from stage 1, bringing it into stage 2, and then proving, hey, we can manufacture 3 batches with data that's within specification, and then you would then submit all of your paperwork to the FDA or EMA for approval. And then also, if you have seen the Kneat portal, there is an article on process validation in there, and you might have seen the term prospective validation. So these first 2 stages are very similar to prospective validation, where you are establishing evidence prior to process implementation based on proposed pre-planned protocols. So you're gaining all of your information in stage 1, and then stage 2, you have protocols that you will then fill out as your batches are manufactured. So do keep that in mind. There's different terms for these different systems or these different stages as well. And then stage 3, this is where you are approved by the agency at this point, and you are continuously analyzing data during your commercial batches, so your real life batches, to show that your process continues to remain in a state of control, And this can also be known as concurrent validation as well, but some of these terms are interchangeable, but this is, again, where you're just continuing to show that goal that you're delivering quality products. So as we go through the presentation, I will take you through each of these stages and go through more detail here, and then we'll also go over some additional information after that. So next slide here is stage 1. So here, this definition up here might look familiar to you. This is the same definition that was on the previous page, so I will bring this up as we go through the different stages just as a reminder. But getting into more detail for stage 1, as I mentioned, this is where research and development will take place, and your quality target product profile is part of that process. So this is where a bunch of tests and studies will be done for each quality attribute of the drug. So if you aren't familiar with quality attributes, this would be things such as appearance of the drug, identity, percentage of water in the drug, different things like that, where you are performing these tests, coming up with a specification range of those quality attributes where the drug is at its utmost quality. So this is a very important document that we will reference in future slides. This is one of the first building blocks for your drug and the different criteria that it should meet to be the best quality possible. And then also in this R&D stage, we have small scale studies here as well. This is exactly how it sounds. It is a study at a smaller scale, or at a bench scale is another term for it. And these studies do have some pros and cons associated with them. For the pros, they do cost a lot less because you aren't using as much of your drug as you would at the larger scale. Your scheduling will be a lot more flexible than working with a CMO, and you have as much time as possible to react to issues. It just depends on your schedule and if the lab is open at that point in time. But also, on the other hand, a lot of the times you have to do some calculation to use that data to scale it up to the actual manufacturing batch size that you will be doing in stage 2 and stage 3. So there is a little bit of a room for error there because it is not a one-to-one comparison and you're using different pieces of equipment. So do keep that in mind with those. And then also a big part of stage 1 are the engineering and clinical batches. So I like to explain these as though they are practice runs that are used to discover and mitigate issues. So with these, it's a really great opportunity to also do large-scale studies, but going into engineering batches and clinical batches a bit more. For engineering batches, these are non-sterile batches. So for example, if there's an issue during the filling portion of the process, the engineers can open up the filler and tinker around a little bit to solve the issue. But for clinical batches, we do claim sterility here. It's treated as though it is a real life batch in stage 2 or stage 3, so nothing can be opened. And because that it is sterile, it can be used for clinical studies. So some of you might have heard that term where they do different clinical studies to see if the drug actually works with the the actual batch and then a placebo batch sometimes. So those batches are often done for those studies. So it might sound familiar to you. And then with those large scale studies, since we're doing these practice runs, it's a great opportunity to also incorporate some studies in there just to continue to gather data on the drug and how it reacts and just gain some confidence before going into stage 2. So on the next slide, we will actually go over a generic process flow that might be seen in drug product manufacturing. And then after that, we'll come back to the large scale studies so you can see how those fit into the process. All right, so here is our high level overview. This again is just an example. This can be more complex than the examples that I have up here or less complex, but this is just an example to help your understanding with where the studies come into play. So I'll just go through the different steps quickly. I won't get into too much detail since it is an example and can vary depending on the client. But the very first step we have here is bulk drug substance receipt, or BDS for short. And over here I have a picture of some bottles. These are Nalgene bottles. And so more times than not, the bulk drug substance won't be manufactured at the same site that formulation and filling will take place, and all these steps are formulation and filling. So the drug has to be received on the site that will perform those tasks, and it could arrive in something such as these bottles or a tank or bag, a lot of different items that it could arrive in, but here's just an example for you to see what it could look like. So it's arrived on site, some tests are performed just to make sure it is the drug that it says it is, because you don't want to continue on with the process with the wrong drug. And then after that, we have thaw or equilibration. So thaw, this means that sometimes the drug is received on site frozen. So it's an ice cube within this bottle. And for obvious reasons, we cannot continue if the drug is completely frozen. So over a couple days, the drug will be checked every so often, every couple hours, to see if there's any ice floating within it until there's no more ice visible. So that would be thaw. Once it's completely liquid, then we'll move on. Equilibration is another option that's commonly seen here, where the drug is not frozen when it arrives on site, but it has to reach a certain temperature before we can continue. So same type of deal, the drug will sit at some sort of temperature, sometimes controlled room temperature, until it hits that temperature that it needs to be at for viscosity reasons, for example. So that is thaw or pooling, and then we get into, thaw or equilibration, and then we get into pooling. So for that, all of these bottles of the drug will essentially be poured into one vessel. So here is an example of a mixing vessel. So all of these 4 bottles will all be poured into one vessel, and that is the pooling step. After that, since we're in a mixer here, you probably assumed that mixing is coming up next. So an example I like to use for this for your everyday life is if you have salad dressing sitting in your refrigerator for a few days and you bring it out to put it on your salad, you probably don't want to dump it on your salad as is. You probably want to shake it up a little bit, especially if it's Italian dressing, because you can see those layers in the dressing bottle. So very similar type deal here. The drug has been sitting for a few days or sometimes a few weeks, and we just want to give it a good mix to make sure the concentration is good throughout, is the same throughout. And then next we have 2 levels of filtration. The first one is bioburden reduction filtration. So I do have an example filtration pathway that might be seen over here. So this vessel will be hooked up to this pathway, and a pump will pump the drug through the 2 filters in series. So this is to get rid of bioburden, or to reduce bioburden, as it says in the name. And for those of you who don't know, bioburden can be live fungi or bacteria, things that you don't want to be in your drug and you don't want to inject into anyone. So we want to get rid of as much of that as possible. And then after that, we have sterile filtration. It's just a quadruple check that everything is out of the drug, all that bioburden. So it is the same type of setup that you see here again. The drug is just fed through it again, and then after that, we have sterile filtered liquid. And then lastly, the filling step is here, where that drug, that tank of the sterile filtered liquid, will be hooked up to a filling isolator, and the drug will be filled into the vials and syringes. And you will actually see a photo of a filling isolator in later slides, so I will point that out. But now that you have seen the overall process or an example of one, we are going to segue into studies so you can see why those are so important and why they need to be done in stage 1. So the very first one we will focus on is mixing. All right, so this is an example of a large-scale study that could be done. And for this, these studies are used to determine the mixing parameters for stage 2, and then also the parameters that you will use for commercial manufacturing. So here we are most concerned about mixing speed, mixing duration, and the associated batch size. So I do have a couple requirements up here that need to be fulfilled in order for this study to have data that you can reference for stage 2. There must be samples taken throughout, and if you do not take samples, you won't have any data to prove that the study was successful, so you need to be sampling and testing those samples. Next thing is the mixing vessel must be the same as the one planned to use for batches in stage 2 and 3. So if you remember back to the small scale study conversation, we mentioned that that wasn't a one-to-one comparison, so there's some risk here. But for these large scale studies, you have no excuse but to use the same piece of equipment for that one-to-one comparison. So very important to use that there. And then also here I have an example of a Kool-Aid tablet being dropped into a glass of water, or you can also think of powdered lemonade being added to a pitcher of water, since summer is coming up and I'm sure we're going to be having a lot more lemonade soon. So with that example, you want to mix the water enough so the powder is consistent throughout. You don't want a sip that's super watery and then concentrated like crazy at the end. So you want it to be consistent, but you also don't want to mix too fast where your Kool-Aid is splashing around and making a mess. So that is very similar to this mixing study actually, is mixing things like you do in your normal life. So I also have a table here that shows example studies that are used in the pharmaceutical world. So I'll just walk through each of these. The first study is where we have the smallest batch size that we mix at the highest speed for the longest amount of time. And this type of study will test shear stress. So typically there's the validation representative in the room when these studies are taking place. So I've seen a great deal of different studies. And when we're in the room, we are looking to make sure there's no splashing, there's no steep vortex in the mixer, and there's no foaming. So if we see any of those things, that is a bad sign, and there might be something going on with the proteins in the drug. They might be breaking down. So if we see something like that, action would need to be taken for that to be reassessed and some of the parameters changed. So that is one extreme there. And then the other extreme is study number 3 that I have up here. So this is where we have the largest batch size that we mix at the lowest speed for the shortest amount of time. And this tests homogeneity. So here we're just asking ourselves, what's the shortest amount of time and lowest speed I can mix at that still adequately mixes the drug, and the concentration is the same throughout. So if you think back to your lemonade, what is the shortest amount of time that it can be concentrated equally throughout your lemonade? So with this study, it's oftentimes seen that samples will be taken from the top, middle, and bottom of the giant batch of the drug with a very big pipette just to test the concentration at those different levels. And then, so we have those two extremes. We have our shear stress study and our homogeneity study. The one in the middle is our target study. So if we can prove that the drug is not affected with study 1 and 3, we are very confident that study 2 will have no impact on the drug. So that's where the target comes into play. It's established through studies 1 and 3, and then that is typically what is used for your actual commercial batches. And typically during the engineering and clinical batches, one of these studies is typically done during each batch, and then these studies will be performed again during stage 2 to officially validate them. But again, we're collecting that initial preliminary data to gain confidence going into stage 2. So that was our large-scale mixing study, and now we're going to go over one more common study at the large scale, that is the hold study. All right, so as you can see here, there's another box here that was not there before for the hold study. So again, this is just an example. This hold study can be earlier in the process or later in the process after filling. But for now, I'm going to put it between the two different types of filtration. So typically filling is a common area where there's lots of delays during the process. So a lot of the times I would see clients have a hold study as close to filling as possible. The filling machine is very, very high tech. There's a lot of robots going on in there. So sometimes if something goes wrong, it takes a little bit of time to fix it. So that's why I did see the hold study oftentimes placed here. Also, you might be wondering why this isn't after sterile filtration. And that is because the FDA does not suggest to do a hold study after the last leg of filtration. So let's say the drug gets contaminated during the study, you're going directly into filling with no extra check for filtration or anything like that. So we do like to have one last round of filtration after the hold study. But that's just an example of where it could be in the process. And now we'll get into more of the hold study details. All right, so a lot of this will be similar to the mixing study. So for hold studies, you are gathering data to determine that absolute maximum hold time between different steps of the process. So you saw that on the last slide. So for example, at a contract manufacturing organization, since many batches for many clients are being manufactured in the same environment, you want to have some wiggle room in case there's a delay due to another client's process or the filling line is down, like I mentioned. So it's good to have extra data in a time of need like that. So typically the worst case scenario is determined, whether that's close to filling or somewhere else in the process that might have delays, and then a validation hold study will be performed. So same type of deal with the mixing study. This would be performed during the engineering and clinical batches, and then that same study will be performed again during stage 2 to officially validate it. So same type of requirements here, samples must be taken. For these hold studies, they're typically done over a series of days. So for this, samples could be taken every 24 hours, every 12 hours for 3 days. It really depends on what we're looking at here and how many samples we want. And then for the tests, typically we'll test for concentration or any type of sterility sample like bioburden or endotoxin. And then of course we want to use the same vessel planned for stage 2 and 3, so we have that one-to-one comparison again. And the photos on this slide, this is an example of a holding vessel that might be used for the study. And then over here, I have a concentration over time graph that you might see in the summary report for the study. All right, so that wraps up our large-scale studies, and we are going to get into more specifics of stage 1 now with the critical quality attributes and process parameters. So getting a little more complicated here. All right, so also during stage 1, you're going to start to determine those CQAs or critical quality attributes and your process parameters. So again, I have a definition up here for CQAs from the Guidance for Industry, but to make it a little simpler, these attributes are essentially measurable properties of the drug that are considered critical for the intended quality of the drug. So I do have some examples up here in that definition, whether it be physical, chemical, or biological. Essentially just the attribute must be within the specified range to be deemed a quality product. So if we think back to our quality target product profile document, a lot of this information and data will come from there. All those tests that were done to determine that specified range that will say the product is at a certain quality when it is in that range, the quality that we would like to have. So for a real world example here, I do have 2 pictures of glasses of water. The one on the left is probably what you would expect to see when you're filling up your glass in your sink or at your refrigerator. And then the one on the right is probably what you don't want to see, and you would question drinking your water and probably would have to make some calls if you saw your water come out like that. So I like to use this example because it's very, very similar to what is done in the drug product manufacturing world, and appearance is a really good example of a CQA that is commonly checked for. So at the end of the manufacturing process, the drug might be checked to see that it's the right color, whether that's clear or yellowish clear, and that there are no additional particulates in it that should not be. So you're checking for some of these critical quality attributes in your own life without realizing it, but this is something that directly pertains to similar situations that are done in the drug product manufacturing world. So that's a little intro on CQAs, and now we're going to see how these quality attributes tie into whether a process parameter is critical or not. So here is a little intro on the process parameters. So to determine if a process parameter is critical or not depends on if variability to that parameter during the manufacturing process could impact a critical quality attribute. And based on what you saw on the last slide, if it does impact a critical quality attribute, that would directly impact the quality of the drug. So that's something we would need to look out for, keep a closer eye on. We don't want that parameter to vary too much because it would impact the drug. So an example of a critical process parameter could be filtration pressure. So if you think back to the flow diagram where we had sterile filtration and BBR filtration, the pressure that the drug is filtered at would be critical. Mixing speed or mixing duration can also be critical. But then on the other side of it, if varying the parameter does not impact the quality of the drug, that would be a non-critical process parameter, or NCPP. So along with assigning whether the parameter is critical or not, we will also assign different values to that parameter, which would be the target value, the NOR, which stands for normal operating range, and then also the PAR, which is the proven acceptable range. So to explain these, I have 3 glasses of water here again, so bringing in the water examples. The first one represents the target, or the NOR. So the normal operating range is typically a range of values, and the target is within the normal operating range. So let's say you wanted to fill up your glass of water halfway. You go over to your fridge, you fill it up halfway, we're good to go. The target and NOR values are also typically what is listed in the master batch record for the operators to follow for the process. So as long as they're within the normal operating range or even hitting the target exactly, we're good to go. The second image is an example of the proven acceptable range. So you filled up your glass a little more than you wanted to, but the volume of the water is still able to be held within that glass. So this is exactly what it says, proven. So you have data to prove that your drug is still okay if you're a little outside of the normal operating range. So this is typically determined through data from stage 2, where you have a little bit of a buffer. If the operators accidentally go outside of the NOR, you'll pull up your validation documentation and say, oh, but our PAR, we have data to prove up to X amount of volume. So we're good to go. The drug is still of the quality that we need it to be. And then the last example with the red X under it is if we go over the proven acceptable range. So if you've ever spilled something in your kitchen, you can imagine how messy this might get. So at this point, we no longer have data to prove that the drug is okay. We're going over the proven acceptable range, and your batch is probably going to need aborted at this point, and you might lose a lot of money. So we never want to go over the proven acceptable range. We definitely normally don't want to go over the NOR either, but that PAR is there just in case as a safety net. And then on the next slide, for those of you who are interested, I do have a table of real-world pharma examples outside of this glass of water scenario that you might see and some values for these different items. All right, so here's an example table, and you might see something like this in a document called the Process Control Strategy, or PCS. Also, depending on your company, you might have 2 separate documents for critical quality attributes and process parameters, or they might be combined into one for the PCS. So here's an example table, like I mentioned, where we have our process parameters, the step where the parameter takes place, whether it's critical or non-critical, and then some values for the target, NOR, and PAR. So you can see that the target is a specified value within the NOR, and then the proven acceptable range is a little broader than the NOR, where we have some data to prove if we do accidentally go above the NOR. So just some examples here for you to see, and this document is one that is regularly referenced if things do go wrong, that we can pull it up and just make sure we're still okay as long as we're in the PAR. So that actually wraps up stage 1. We're going to get into stage 2 and 3 now. There will be a lot less information for those stages because stage 1 is the big one where you're gathering all of that information and data to go into stage 2, but we'll go ahead and segue into the next stage. All right, so to set the scene a little bit for this, at this point we have gathered all of our data from stage 1, and we're ready to start with our process performance qualification batches in stage 2. So I have that definition up here again, and we'll get into more of the details. So the first bullet point is equipment, utility, and facility qualification. So some of you might be thinking that this is a little bit of a gray area and could potentially go under equipment validation, which is very true. There were only 2 equipment-related items that I worked on in my previous position, so I'll just touch on those because it is kind of a gray area, but there's a lot more equipment qualifications that could go on depending on if the equipment is client-specific or not. So the first one that I had experience with was the component compatibility operational qualification, or CCOQ for short. I know it's a mouthful, so more times than not we call it CCOQ. So for this, this will take place if there's new components that are being ran on the line for a project that hasn't been used before. So I won't get into too much detail here, but you will be running those pieces, those components, whether it's vial stopper seal combo or syringe and stopper combo, on the filling line just to ensure that it runs properly. And here is an image of a filling isolator. You can't really see all the robotic arms going on in there, but if you have some spare time to look up a video for a vial filler or syringe filler, I definitely would recommend to do so. So that's the CCOQ. And also an additional item that goes hand in hand with that is container closure integrity testing, or CCIT. And this just ensures that the unit is accurately sealed and nothing can contaminate the drug in the unit. So we'll go through a series of testing for dye ingress and microbial ingress testing there, just to make sure nothing can contaminate the drug, because we definitely do not want that. So that is, again, can be determined to be equipment depending on the company, but those are some items that you might see. And then process performance qualification. This is the big one for stage 2, and this is where the client will manufacture 3 different batches as though they are commercial or real life batches with a bunch of additional testing on top. So if you think back to that flow diagram again, you'll go through from BDS receipt all the way through filling, and you will have a bunch of different samples and testing done throughout to prove to the FDA that you can deliver consistent quality products, back to that goal. So for this, you will have some documents that you have to complete. You'll have a PPQ protocol where all of your tests and samples will be recorded, that pre-planned protocol. You will also have a PPQ readiness checklist that will assess if there's any outstanding change controls or deviations that could impact the PPQ batches. If there is, a risk assessment would also take place there, and it will assess that all your documents are effective, things like that. It's a pretty long checklist. And then also, those of you who have validation experience, where there is a protocol, there's always a summary report. So there will be a summary report authored after the 3 batches are produced. And oftentimes that same summary report is used to submit to the FDA or whatever agency, and used to prove that goal that they're able to continue to manufacture that drug without any issues. So really cool stuff there, where we're getting into the actual qualification. And then also something that is oftentimes overlooked at this stage is ongoing studies. So this is where I'll dabble into drug substance just a little bit, where you could have resin reuse studies or resin life cycle studies. So these are studies that would be going on in the background of a commercial batch, but it would not jeopardize the batch in any way, just to continue to gather information to improve and better their process. Just because pharmaceutical things are very expensive for those of you who are in the industry. So any data to make the process better is definitely what we want to have. So ongoing studies can also occur during stage 2. That actually wraps up stage 2, so we'll move along to stage 3 now. All right, so setting the stage for this stage, I guess I should say, this is where you are approved by whatever agency you filed with, and you are going to continue to prove that your process is in a state of control, where you're going to continue to gather data points for your critical process parameters or non-critical process parameters and analyze those different data points statistically. So very much like you see on here, you might have many tab charts with those different points with the high limit, the low limit, to analyze what is within specification and what is out of specification. So some documents that you might see here is a continuous process verification plan, where if you are working with a CMO, there's typically a plan in place for when they would like to have a continuous process verification report. This could be after 10 batches are manufactured or 20 batches are manufactured, but typically there's an agreement on when they would like to see a report and analyze that data. And then also there are periodic reviews, which happen on a yearly basis. So if a client wants to see how their batches did throughout that year, any deviations that were opened, they can see all of that in a very nice report with all the summarized data as well. So this is a very, very important part of the process, as are all the other stages we went through. But you do want to keep an eye on those critical process parameters and make sure they're within specification and any other parameters that you would like to analyze as well. So also at this stage, you are given the opportunity to look at the parameters. If there's one that's continuously out of specification, you can assess it, determine the root cause of why it continues to be out of spec, and then also come up with a path forward on that. So we're getting into the corrective action, preventative action type deal there. And for example, if we go back to our hold times that we talked about, if there's one hold time during the process that you keep exceeding time and time again and deviations keep needing to be opened, you could go back to stage 2, do a couple hold studies there, and then validate that new hold time and then minimize the deviations in the future. So you have that flexibility here to improve your process. And also, I do want to emphasize how important it is to ensure your critical process parameters are within spec. So if something is out of specification, your batch could be aborted, like we mentioned, if you go over par, that proven acceptable range, which could lose a company a lot of money. And if a batch is released for human use and later on it's found that a critical process parameter was out of spec for that batch, this could lead to product recalls, and then companies' names could be ruined forever. So I'm sure you have seen some things in the news over the years where something like that happened, and then you find yourself not really wanting to use that company's products anymore because of what happened. So there's big risks involved, which is why it's so important to keep an eye on every single detail within this. So difficult to keep track of, but it's very, very important to put in that work, because later on you might have some issues that arise that you don't want to deal with at that point in time. So that actually wraps up stage 3, and there's one more slide on revalidation that we'll quickly go through, and then we'll finish up with our case study. All right, so revalidation, this does tie into a little bit of what I discussed for stage 3, but if there is a major change to your current validated process that you went through stage 1 through 3 with, revalidation will most likely need to be completed. So oftentimes for this, it means that an addendum PPQ batch will be manufactured, which is just one confirmatory batch that will be compared against your original PPQ, and just ensure that everything remains the same between those two batches. So I do have some examples here on when this might be needed. So the first one, if there's a change in component, so you might need to redo a CCOQ or CCIT here, depending if that component has been previously used. And as you can imagine, the drug directly touches that vial or that syringe. So we'll do an addendum PPQ with focus on the filling section of that flow diagram, just to make sure nothing is impacted. And then also the next two are similar, they're equipment related. So if there is an increase in the batch size where you need a new piece of equipment, or a change in equipment where it's not like for like with that original piece of equipment, typically an addendum PPQ would also be done. And then the last example is a change in the drug's substance supplier. So you might be thinking that it could be argued that the drug would be exactly the same. It's the same batch record, just a different supplier and a different site. But in reality, a new supplier can impact CQAs and CPPs. So we must do that one addendum confirmatory batch to just make sure that we are all good. So now we will segue into the case study that is done by Merck, and we'll go through how their process was before Kneat and then after Kneat. All right, so for those of you who do not know Merck Sharp and Dohme Corporation, or MSD, they are a pharma company who produces biologics, medicines, and vaccines for a wide range of diseases. They are a pretty big company, so I'm sure most of you are familiar with them if you're familiar with big pharma. So this case study includes process validation as well as some other validation disciplines as well, which we'll get into. And the main takeaways from this case study are that they saw a 50% cycle time reduction in execution of testing, and also 27 sites were rapidly deployed to use Kneat. So all this data is taken from those different sites, so wide range of data, and of course we love to see that 50% cycle time reduction as well. So on the next slide, we will take a look at Merck's process prior to Kneat. All right, so prior to Kneat, Merck did use a mixture of paper-based and digital validation approaches. So as you can imagine, this did lead to some difficulties and challenges that they faced. So when you are using several different systems, it's not the same avenue or way that they use to create a proven number documents. So the documents that they were using and working on were not consistent with all these different paper and off-the-shelf systems. And then also, as you can imagine, if you have been involved in a review process before with different comments, since there were different systems, it was difficult to consolidate all those comments, and also over communications had to be done between the authors, reviewers, and approvers, just to ensure everyone was up to date on the most recent documents and the most up-to-date documents. So that can be a challenge. And then of course, I'm sure everyone's aware of the giant binders of all the validation paperwork, and keeping track of those can be difficult if everything is offline and it's paper-based. So ensuring that those binders remain in a safe, regulated place can also be difficult to do. And then lastly, we'll take a look at Merck's process after Kneat and what changed. So Merck chose to digitize 7 different validation processes across 27 sites using Kneat. So as I mentioned, this did include process validation. A few other disciplines were CSV, cleaning validation, supplier qualification, a bunch of different disciplines there. And through the use of Kneat, they were then able to generate, number, and approve those documents in one system. As you saw on the previous slide, there were multiple different systems in use. They were able to collect those review comments in one system as well. And then through Kneat, it does notify the author, reviewers, and approvers of when they need to take action on a document. So they now have that automatically generated system using Kneat. And then also all their documents were then electronic after they started using Kneat. But also these documents can be exported out of Kneat if you would like to see the paper-based version as well, but they are all organized in the Kneat workspace. So some highlights I wanted to touch on is that 50% cycle time reduction per testing that we love to see. Their process steps were simplified and minimized from 15 to just 8. And then several systems were used before Kneat, which made things complicated, but they were able to minimize the number of systems they use from 5 to 2. So as you can see, many benefits of switching to Kneat, just with reducing the time, which then is a lot less money to do things. And just all the highlights here is just really great to see for those of us who work at Kneat and for those of you who might be interested in using it in the future.
Wonderful. Thank you so much, Olivia, for that detailed presentation. We will get a few questions, but before we start, I did want to just make an announcement that we have officially launched this data validation survey. Essentially, this data validation is an annual validation industry report powered by Kneat and our partners, and it aims to shed light on practices, progressions, and pitfalls of the validation industry globally. So if you complete the survey before the end of June, you will be able to get a $20 Starbucks gift card and also enter it into a raffle to win an online Kneat Academy end user certification course. So you can scan the QR code on the screen or head to stateofvalidation.com/survey. It's about a 15-minute survey, and we would appreciate as many responses as possible to get a really good collection of data. And you will also, if you do fill in the survey, you will also be on the list to get the authored report first once we have all of the data consolidated. So exciting times. So head to that website to get into that survey. All right, I know we're a little bit out of time, but I will maybe just save time for a couple questions. As mentioned at the top of the program, we do have this presentation recorded, so you will get an email later this week with the slides and the recording, so you can always go back and reference it. And if you have other questions that come up later, you can always email info@kneat.com. But the first question I see here coming in, Olivia, is it common practice for there to be 3 PPQ, the process performance qualification batches manufactured during stage 2?
Sure, that's a really good question. So it is common for there to be 3 batches, but also there was a recent change a little bit in the regulations where if you can prove that your process is in control without doing 3 full batches, that is something that can be done now. So if you've done a lot of different studies during stage 1 and you feel very confident, you can do less than 3. And then also on the other side of that, you can do more than 3 if something happened during one of the batches and you have to do a redo, or with the addendum batch, that would be 4 batches with the change. So 3 is definitely the common practice, but it's not uncommon to see less or more at this point.
Perfect, thank you very much. There was also a question about just the exact 7 work processes that Merck was able to digitize in Kneat. So I'll just kind of repeat those ones again. It was the computer system validation, facilities, utilities and equipment validation, process validation, cleaning validation, equipment sterilization, laboratory equipment validation, supplier qualification, and the validation master planning. So a very long list there, but it does kind of show functionality and capabilities of the Kneat platform that way. All right, okay, one more question and then I will wrap up as I do want to be mindful for people's time. From your CMO background, how did you go about determining if a process parameter was critical versus non-critical with the clients you worked with?
Okay, yes, so for that, since I worked on the technical transfer team, the client would send us a lot of their information that was done during stage 1 for research and development and any small scale studies. So typically when they came to us, they already had an idea in mind of what was critical versus non-critical. So I would take a look at those and we would have a meeting to go through each of the different process steps and the parameters. And if there was something that we weren't in agreement on, whether it should be critical or non-critical, we would have an open discussion on that, and then we would come together with a conclusion at the end. So definitely lots of discussion around that, because since they aren't actually on site, they don't have all that information on what is actually non-critical when they might think something is critical. So lots of discussion, and we come together in probably an hour or 2-hour meeting to discuss those and come to a common ground.
Perfect. Thank you so much. All right, well, we have come to the end of time here. Thank you everyone on the line for joining today's presentation. We really appreciate you tuning in. And Olivia, thank you so much for taking the time to prepare this information and share your knowledge with the audience.
Of course. Thank you everyone for joining. Hopefully you learned something today.
Absolutely. Thanks so much, everyone. Have a great day.
Bye. Thank you.